Okay. So
Yeah, okay.
feel free to bring doubts. Okay, so I don't know if much time it will take her to join. Anyway, so let's go with a quick summary of what we did in the last class. Where did we start in the last class?
Skeletal muscle.
Skeletal muscles. Yes, right. Cool. So let me start with the revision, okay, with the summary, and then if Devangi joins by that time, she'll pick from there. Otherwise, would you like to do the rest of the half?
Yeah, okay.
Yep. Okay. So we discussed about skeletal muscles, where we first talked about the structural details of skeletal muscles, and as we know that skeletal muscles are also known as striated muscles, or and they are voluntary muscles. They are they are under the voluntary control of our central nervous system. So you see that they are arranged in a manner where one muscle bundle is called a fascicle. Okay. And it contains fascia, which is collagenous, and it keeps all the muscle bundles together. Okay, so that's why it's called fascicle. And inside the fascicle, you will find muscle fibers, individual fibers, which are also each muscle fiber is like a cell, is a myocyte, which is elongated. Okay, and these fibers are covered with sarcolemma, and this sarcolemma is like a plasma membrane of these muscle fibers, which contains the sarcoplasm, which is again multinucleated. It's like cytoplasm. So here it's called sarcoplasm. And then muscles are always in present or is feeded by blood capillaries, because blood bring nutrition and takes waste away from these respiratory waste away from these cells. Okay, so multiple such bundles make a whole muscle uh, muscle what do you say, a bigger muscle chunk that we see, right? So that's why if you see any cooked flesh, cooked muscle, it will look like thread like structures together. So these threads are basically these fascicles. Okay.
Yes, sir.
Yeah. And then we went on to talk about this muscle fiber is a syncytium, which means um, it's a continuous cell where multiple nucleus can be present. And then we talked about myofibrils, which are muscle filaments, which gives rise to this dark and light bands in these skeletal muscles. That's why they are called striated muscles. Okay, then we talked about these filaments. There are two kinds, actin and myosin. So I think Devangi has joined. Devangi, we are waiting for you. Would you like to summarize from here the actin filaments and the myosin filaments? Am I audible to you, Devangi?
Yes, sir.
Yeah. If you're speaking, you're on mute, Devangi.
So it's structure of actin and myosin, right?
Yes, from here, what you see on screen. So how do you understand this figure? What is this all about? What is I band and all this?
Yes, sir. Myofibrils are muscle filaments arranged parallelly in the sarcoplasm, and they have dark and light bands on them due to the presence of actin filaments and myosin filaments. These are the actin and myosin are the proteins. Actin filaments are called light band, and myosin filaments are dark band. So actin is attached by Z line in the I band, and A band is the dark band that is made up of myosin filaments.
A
One
also has
my
overlapping actin. Right? A band also has some overlapping actin.
yes, so one myosin interacts with two actin filaments,
Mhm.
and in H zone only myosin is present, whereas in A band both myosin and actin are present, and in I band just actin.
Correct.
And between one Z line and another Z line, it is called a sarcomere.
Yes, correct.
Then structure of actin and myosin. Each actin filament is made up of two strands of F-actin, which are helically bound to each other, and another two filaments of tropomyosin is associated with the F-actin. So the protein troponin is present at regular intervals on this tropomyosin protein, and in resting state, troponin covers the actin binding site for the myosin. And myosin is a polymer protein. In the head region, there are two sites, one ATP binding site and another actin binding site, and there are two heads.
Correct.
Then we have mechanism of muscle contraction. So actin are the thin filaments and myosin are thick filaments. So the sliding filament theory states that contraction of muscles or myofibrils takes place by the thin filaments actin sliding over the thick filaments myosin. So the signal for contraction of muscle comes from the central nervous system through a motor neuron. This motor neuron forms a neuromuscular junction with the sarcolemma and releases neurotransmitters, acetylcholine.
Right.
Then this neurotransmitter release will generate action potential in the sarcolemma, which spreads throughout the muscle fibers.
Yes.
So then we have calcium ions in flux. That means they move into the sarcoplasm, and there they bind to the troponin and remove it from the active site of myosin on actin filaments. So then myosin head utilize energy from ATP and bind to the actin.
Right. And this is where the cross bridge formation takes place, correct?
Yes, sir.
Yeah, perfect. Thank you so much, Devangi, for this beautiful summary. Now I'll take over from here. So today we'll take from here, and in the last class we reached till this cross bridge formation where you see this myosin attaching to the actin at sites where the sites which were previously occupied by troponin. So now this troponin is gone. So because calcium came and bind to troponin and takes troponin away. So now these sites are available for this myosin binding. So myosin heads bind to that site. So the bridge is ready. Now you all have, both of you have NCERT with you. Can you come to the figure 20.4 in your NCERT? Everyone? Yes, Devangi is there. Chania, what about you? Come to the figure. I'm also trying to pull that figure out. No, that's not it. Yes, that's the figure. This figure I'm talking about. Can you see this, everyone? Both of you? Yes. So story from here, further on what happens. So what you can see here is a cross bridge formation. Okay. So this is where we start. This is hydrolysis. So this is where there is a cross bridge formation, right? You can see cross bridge, right? This is what we just talked about. So when there is a cross bridge, it's like it's forming a 90-degree angle. Okay, do you understand? Can you see this, everyone? So
Yes,
here,
sir.
there is a 90-degree angle between the myosin head and the actin filament which is present. Now what happens after this is that this binding, so due to this binding, the attached actin filaments are pulled towards the center of the A band. So now we will keep going towards to this thing. So now imagine, let me take this image also there. So yeah, it will be easier to keep it here. Yes, correct. Yes. So now imagine, so now myosin has bound to actin, and this is where, so this is myosin. Can you see these points? These points. So these are the points where these myosin heads are poking out. So there will be multiple places where this myosin will bind to actin because they were troponin. Remember, at particular places, at regular intervals, there were binding regions for myosin on the actin filament. So this kind of binding will happen here and also will happen on the opposite side, because I told you then one big myosin filament, like these are group of myosin filaments, they will bind with the actin filament here. This part is clear. So let's assume that all of them are under cross bridge formation. Now this is the starting of our muscle movement. Now when they will be formed, first this is, let's say, step 1, like position 1. From position 1 we go to position 2, where the sliding happens or the rotation happens. So the myosin head rotates, moves towards its this tail region. So this head tries to come this way. Okay, that's called the rotation of this myosin head. And because of this rotation, the actin filament on the top will slide in this direction. Is it clear? Thumbs up if this is clear. And now look here, when this actin will slide in this direction and this actin will slide in this direction, right? Because you can understand in the orientation of binding is opposite. So what will happen? This A band will get shortened, shrink, because actin now comes into the A band, that it slides over myosin and narrow down the A band or closes the A band. Is that clear, everyone? Yes. So write down the first point. The binding of myosin head to the actin filament. The binding of myosin head to the actin filament will pull the attached actin filament, will pull the attached actin filament towards the center of A band. Towards the center of the A band. Now, what are these actin filaments attached to, Jania? What are these actin filaments attached to? Can you see in the figure they are attached to something? The figure below, which shows striations. Can you hear me, Jania? Are the actin filaments free, Devangi?
So they're attached to myosin heads.
No. Apart from myosin heads, when myosin head was not attached, was it floating in the air? Myosin head just attached. We just saw the attachment now to make a cross bridge. Before making a cross bridge, when the troponin was sitting on actin, myosin could not bind to actin. Where was actin attached? Do you see in the figure actin is attached somewhere? To keep in place, you have to attach it to something. You only told.
Z line.
Exactly. It is attached to the Z line. Z line is nothing but an attachment protein. Okay. It's not a line. It just looks like a line, a darkly stained line in our muscle staining procedures. But this must be, of course, it is another protein that is called the attachment or scaffolding. So it acts as an attachment protein for actin. So it is attached to the Z line. Now my question to you is, what will happen to the Z line in response to this pull? So we realize that when myosin will attach to actin, it will pull the actin filaments towards the center of the A band. Okay, cool. But what will happen to the Z line? They are attached to Z line also. Logically, what do you think will happen? Look here. This is the A band. Can you see here, people? And what is this? This is the Z line, right? And this is an actin which is attached here. And that is myosin. Now myosin pulls A towards the center. So this A line will close up. But what will happen to this Z line? Actin cannot stretch like a rubber, no? So this Z line will also be pushed inside. Do you understand? Is it clear?
Yes.
If you have holds onto something, let's say you are holding the door and I'm holding you and I try to pull you, then you will create a pull on the door. Correct? And the region between two Z lines is known as?
Sarcomere.
Exactly. So when myosin head will bind to actin filament and will make that actin filament slide over and come to the A region, what will happen? The length of the sarcomere will shorten. Each sarcomere will become shorter. Correct? Yeah. And that is exactly what we know as muscle contraction. This is how muscles contract. So muscles contract, write down. Next point, the Z line, which is attached to these actins, the Z line, which is attached to these actins, will also be pulled inwards or pulled closer, will also be pulled closer, leading to shortening of the sarcomere. Leading to shortening of the sarcomere. Also known as muscle contraction. Also known as muscle contraction. Is that clear? Devangi and Janya? Good.
Sir,
Yes. Yes, Devangi.
sir, if the Z line moves inwards, then what will happen to the other actin attached on the other side of Z line?
Exactly. So that's the next thing that I was going to talk about. So there will also be a pull on the other actin filament, right?
Yes, sir.
Yeah. So the I band, you know what is I band, which is just actins. That is I band, right? So the I band itself will get reduced. So the other actin, depending on in which direction you want the muscle to contract. So for example, in our biceps, when we flex or when we shorten the arm or close the arm, the biceps get contracted and hence there is a bulge in the bicep, right? So the muscle are contracted and coming together and making a bulge, right? So this whole I band reduces. So in a muscle contraction, you will not see this I band in a contracted muscle. You know, this band you will not see. After Z line, this whole area you will see will be darker, because here also this I band is being pulled and it will, this darker region will extend like it is extending here. Do you understand, Devangi?
Yes, sir.
Yeah, so write down. In muscle contraction, this was the next line. In muscle contraction, the I bands get reduced. The I band gets reduced. So I band gets reduced, whereas the A band, sorry, I labeled it as A. This is actually H zone, right? I'm sorry. This is H zone. A is the full from here to here, right? So the H zone will reduce, the I zone will reduce. What will happen to the A zone, the A band? Can you tell me, based on logic, Devangi and Janya, what will happen to the A zone, this A band? Let me erase and make it clear for you. What will happen to the A band? This, the A band is where myosin is present. So is myosin contracting on relaxing? Is there any pull on myosin itself? No, right? Myosin is the one which is making actin slide over itself. Do you realize this? So myosin is making actin slide over itself. So during contraction, what will happen to the A band? A band will retain as it is. The length of A band will not change. So write down. During muscle contraction, I bands get reduced. I bands get reduced, but the A bands retain the length. But the A band retains the length, because A band is the length of the myosin. Okay, not the actin. So every all the bands which are associated with actin will reduce during muscle contraction, but the A band, which is related to myosin, will stay as it is, and the H zone, which is the gap between two actins, where only myosin is present, will also reduce, right? Because of the pull. Does it make sense, Devangi? So
Yes.
Z lines come together, I band reduces, H zone reduces, and sarcomere shortens. But the only thing which stays the same is A band. So for a contraction of a muscle also, there is a limit. It is limited by the A band. It cannot go smaller than A band. So muscle contraction can happen, like it can contract this much, right? On both the sides. The moment it will touch this myosin, do not contract too much. There can be slight contraction, but the A band will stay. So the maximum contraction that can happen is this much on this side and this much on this side. And that is enough, right? Upon contraction, muscles don't become so short that they just become half of their size. They just contract and then relax. And this happens very fast in a rhythmic manner. So write down. So what happens next? Till this point, it is clear. Till point 2. Everyone? Very good. Now from second, we go to the third step. And you can see what's happening here. During this sliding, the ATP, okay, is broken down into, sorry, write down this point. The myosin, when the ATP is used up, okay, so the energy of ATP is used up here, and you get one ADP and one P. It's called PI. So we always in biology write it as ADP plus PI. I means inorganic phosphate. So when ATP is hydrolyzed, it becomes ADP and PI. So when this ADP and PI is formed, these get released because they cannot stay bound to the myosin heads. Because remember, there was only the site for ATP binding. So the moment you break down ATP, it cannot stay bound and it has to go away. So it goes away. So when this ADP and P goes away, the cross bridge, which was formed upon which the sliding was happening, gets broken, and because the muscle has elastic properties, the myosin head comes back to the original position, how it was in the beginning. Okay? Is it clear? So write down. The myosin, after utilizing the ATP, releases the ADP and PI, releases the ADP and PI inorganic phosphate, which was the third phosphate. ADP and PI, and goes back to its relaxed state. And goes back to its relaxed state. Okay. Write down, a new ATP binds Then again, you need a new ATP, which you can see here. A new ATP binds and the cross bridge is broken. A new ATP binds, the cross bridge is broken, and the ATP is again hydrolyzed or broken or utilized. You write utilized. The ATP is again utilized to the ATP is again utilized by the myosin head and the cycle of cross bridge formation repeats. The cycle of cross bridge formation repeats. Is that clear? Okay. And this process, till what it will continue? So what are the limiting points? So if I start running and keep running, till when I will run? When I'm tired. So what happens when we are tired? Till which point muscle can keep contracting and relaxing? Till there is two things. One, energy, which is ATP, because myosin and actin will always be there, right? So what are required for this activity to happen? One is ATP. So if you are run out of ATP, which means you are exhausted of energy, there is no energy in your muscles, then you will just fall down. None of your muscles will be able to move. But that rarely happens. When we stop moving, this contraction also moves. So normally, if you stand up from your place, move to the other room and sit there, what has now stopped? Is ATP not present around? It is still present. Then why does this whole cycle stops? In a normal condition, what makes this cycle or what makes muscle movement stop? The answer lies in the above paragraph that I dictated you. Can you figure out? How can you make this cross bridge formation, sliding, cross bridge breaking, coming back to original state, again cross bridge formation, then again sliding to stop? How can we do that? Jania? Devangi? Am I audible, Devangi and Jania? Hello.
Sir, if tropomyosin
Okay, let me give you a hint before you try. I think you're going in the right direction. The hint is these muscles are voluntary. And your brain, you can decide when to start contracting and relaxing them and when to stop. So if you want to move your leg muscles and walk, then you can do that. Then you sit and you decide, now I don't want to move. So you sit. Now this is the hint. So what can you change where this whole thing stops? Yes, Devangi. Now try. You cannot.
So acetylcholine will not be released.
Great. Okay, cool. So what will happen? So you are saying that you will stop your motor neurons signal giving to the muscle, right?
Yes, sir.
Okay. So, but when you stop doing that, what will happen to the muscle? What are you doing? What are you changing in the muscle? You are changing the calcium influx. So this can only happen till a point where calcium is coming inside the cell and it is binding to troponin, removing troponin from its site so that myosin head can bind, right? So if I want to at will stop all of this from happening, I will stop signaling from neurons to muscles, which will lead to stopping of calcium influx. So there will be no calcium inside. So if there will be no calcium ions available, no calcium ion will bind to troponin, and then troponin can go back and bind to the this actin filament, right?
Yes,
Correct.
sir.
And when it will be bind, then myosin cannot bind. So there will be no cross bridge formation. And if there will be no cross bridge formation, there will be no sliding. If there will be no sliding, there will be no contraction. If there will be no contraction, there will be no relaxation and there will be no movement at all. Next time when I again want to start muscle movement, I will just send signal from my brain to my muscles that, okay, let's move. And acetylcholine will be released. That will lead to a lot of calcium going inside the muscle cells, binding to troponin, removing it from the actin filament, which is called unmasking. And because of that unmasking, myosin will bind, cross bridge will form, sliding will happen, and muscle movement will start. Does that make sense? Yep. Janya, does it make sense to you? Very good. So write down this process can only continue till the calcium ions are present inside the muscle cells. This process can only continues till the calcium ions, Ca2 plus, are present inside the muscle cells. The muscle contraction can be stopped. The muscle contraction can be stopped by stopping the motor neuron signal. By stopping the motor neuron signal to the muscles, which leads to prevention of calcium influx or stoppage of calcium influx. Due to which the masking of actin filaments, due to which the masking of actin filaments by the troponin takes place. Okay. And write down, this will cause this will cause the return of Z lines. This will cause the return of the Z lines back to their original positions. Back to their original positions. In the bracket, write down relaxation of muscles. Relaxation of muscles. Okay, is that clear? Everyone, Devangi, Janya. Very good. Now look at this diagram. There's one diagram 20.5 in your book. And there is a paragraph, I think, below it, just one paragraph. Can you both see figure 20.5? Devangi and Janya. Okay, so Janya, would you, okay, so before that, I give you 2 minutes to look at figure 20.5 and see if you understand it. Let me see if I have the figure, I can pull it down. Yeah, I have this. This figure I'm talking about. Can you see this figure? Yes, so I give you 3 minutes. Look at this figure and tell me what's happening here. So I'll go to Janya first. Janya, you have to explain this. Based on everything that I have told you, it's just a summary of that. So you have to tell me where in figure, what is this figure telling you? So take 2-3 minutes and we'll start with the explanation. Okay, let's start, Janya. What do you understand by this figure? You will speak and I will annotate with you. Am I audible? Are you there, Janya? Devangi, am I audible?
Yes, sir.
Okay, so would you like to tell us? What is this figure telling you?
Yes, sir.
Yeah.
So in the first phase, the muscles are relaxed. So we can see that H zone and I band are as it is.
Mhm.
Um, then in just below that, the
A band
muscle
is also
has,
as it is.
yes, sir.
And there is this is the Z lines have a longer distance between them.
Yes, sir.
Yes. Then
Then when the muscle is contracting, so as we can see, the distance between the Z lines have reduced. So the length of sarcomere has reduced.
yes.
And also the H zone has shortened as compared to the above figure.
Yes.
Um, and also we can see the shortening of I band.
The I band is not there. I band is gone. Like almost gone. Can you see? It is just
Yes, sir.
this much. And what about the A band?
And the A band is of the same length.
Yes, this is A band and this is of the same length. Correct? Perfect. And then what is the third one?
Then the muscle is contracted to its maximum.
Yeah.
So I band has completely disappeared
Yes.
and
No I band. And
H zone is also disappeared. The actin are overlapping.
yes, so H zone is not there. Instead, there is a overlapping in the actin.
And the Z lines are also almost touching the myosin. So
Exactly. But what
the
happens to the A band?
A band remains the same.
Exactly. A band, so I told you, no, that the maximal contraction can also happen, and that will be the maximum length of the myosin in the sarcomere. So two sarcomeres come so close together, which is their maximal limit, which means the myosin is touching the Z line. Correct? It cannot contract beyond that. Myosins cannot
Yes, sir.
contract beyond that. So even in a maximally contracted position, what stays the same?
A band.
A band retains its length even in maximally contracted position of muscle. Okay. Is that clear, Jania? You have any doubt? Okay, it's clear. Okay, so Devangi, would you like to read the paragraph that I was talking about just beneath it? Read it from the reaction time. There must be a word reaction time.
Yes, sir.
Yeah, the fourth line, I think.
The reaction time of the fibers can vary in different muscles.
And why
Repeated
is that?
activation.
You understand this point? The reaction time means how fast you can contract and relax a muscle. It varies in different muscle types. For example, in our legs versus in our fingers, maybe we can move our fingers faster than our legs, right? Or in our neck, for that matter. So that reaction time can vary. Yes, continue.
can vary in different muscles.
Mhm.
Repeated activation of the muscles can lead to the accumulation of lactic acid due to anaerobic breakdown of glycogen in them, causing fatigue.
Yes. That's where the muscles are tired.
Muscle contains a red-colored oxygen-storing pigment called myoglobin. Myoglobin content is high in some of the muscles, which gives a reddish appearance. Such muscles are called the red fibers.
Correct.
These muscles also contain plenty of mitochondria, which can utilize the large amount of oxygen stored in them for ATP production.
Yes.
These muscles, therefore, can also be called aerobic muscles.
Yes.
On the other hand, some of the muscles possess very less quantity of myoglobin and therefore appear pale or whitish. They are called white fibers. Number of mitochondria are also few in them, but the amount of sarcoplasmic reticulum is high.
Just
They
like there is a
depend.
cytoplasmic reticulum or endoplasmic reticulum in normal cell, there is a sarcoplasmic reticulum in muscles.
Yes, sir. They depend on anaerobic process for energy.
So remember I told you about white muscles, white fibers and reddish fibers or yellowish fibers. Big muscle fibers like ligaments and tendons, right? So it's talking
Yes,
about
sir.
that. So the red fibers have more myoglobin, which is a oxygen carrying molecule. So it's red. It's a counterpart of hemoglobin, which is present in blood. So myoglobin is present in muscles. So these muscles do aerobic respiration to break down, use oxygen to break down glucose to produce ATP. On the other hand, there are white fibers which has less hemoglobin, so less oxygen. So they mostly depend on anaerobic processes. Now, these muscles, both of these, over time, when they are excessively doing this contraction and relaxation, in aerobic muscles, if there is lack of oxygen, if in anaerobic muscle there is no oxygen, anyways, no problem because those are anaerobic. But aerobic muscles, if there is lack of oxygen, then lactic acid starts producing and depositing, and that starts paining in the muscles. Okay, is that clear? Okay, great. So this till here, I want you to take a look at everything that we have done. So all the concepts for this chapter are done. So this was the major concept of locomotion and movement. And now, if you have any doubt, please ask. So I will stop here for a couple of minutes to take your doubts. After that, I will be projecting the NCRT and we will go over the factual part of this chapter, which is skeletal muscles, diagrams and things that you have to know. So instead of bringing every diagram here, I will just project the NCRT and we will discuss through that. And at the end, there will be disorders of muscular and skeletal system that we will talk about. So is there any doubt? Please take a look, and I'll just stop here for a couple of minutes and I'll be back. Okay. I need to get some water. Is that okay, Devangi and Jania? Yep. Okay. Hello. Hello, Devangi and Jani. Am I audible?
Yes, sir.
Yes. So, any doubts in any of the concepts of muscle contraction? Devangi? Jani? Any doubts so far? No. Okay. Okay. So this was about the muscles where we had some concepts of muscle contraction. Now the second part of this chapter is for the skeletal system, because skeletal system is the system that is made up of the bones. And as we know that bones by themselves do not move. Okay, they can only move through muscles. So that part is more factual. So I will stop sharing this thing and let me start sharing the screen, this chapter. Can you all see this screen that I'm sharing? Chani, Devangi? Yes, great. Cool. So let me this part here and let's go quickly to, we've covered all this, yes, to the skeletal system. Now just pay attention and I hope you can see my cursor, right? Can you see my cursor, of you? Okay, cool. So coming to skeletal muscles, what we'll do is, because it's factual, so I will be taking you through the diagrams and we'll be talking about some important things that you need to know. So you keep your books open with a pencil in hand and keep underlining the important things that you have to focus on for your boards and entrance. Okay, you don't have to remember each and everything by heart, but some important things you should remember. For example, the skeletal system, it basically is made up of bones and cartilages. Now, what is a cartilage? If you remember quickly, let's discuss this. What is a cartilage? Devangi? Any idea what is a cartilage?
It's there at the end of bones.
You're right. They are sometimes present at the end of the bigger bones or the joints, right? But they're also present like our ears. So the softer parts, which still needs a shape, but you don't want to put a bone there. For example, your ear is made up of cartilage, the outer ear, right? The nose, the nose is half cartilage. You can bend it, right? So cartilage is agile and flexible, not as hard as a bone. And it also makes structures like supportive structures in our body. It's also present at the ends of bigger bones like our joints of the knee and your right. Okay. So without a skeletal system, we won't be able to move because there'll be no shape of our body. Without the jaws, we cannot chew, and without the limbs, we cannot walk or hold anything, right? So bone and cartilage both are specialized connective tissues. So they are tissues, okay? Just like muscle tissue and a blood. So blood is the only liquid tissue. Bone and cartilage are connective tissues, okay? Bone has a very hard matrix due to calcium salt. So mark this point. Whereas cartilage has less harder, a pliable matrix due to this chondroitin salts. So these two words you have to remember. In bone, you will mostly find calcium salts. Okay, calcium phosphate, calcium carbonate. In cartilage, you will not find much of calcium salts, but you will find chondroitin salts. Okay. So bone are called osteo. So you must have studied the chapter where we were discussing about animal kingdom. So there were two kind of fish, bony fish and cartilaginous fish. You remember what was the name of the bony fish, scientific name of the bony fish? Devangi? Bony fish were known as osteochthyes. Okay. And
Yes.
cartilaginous fish were called chondriochthyes, right? So the word chondro is associated with cartilage and the word osteo is with bone. Now you just know there are 206 bones and a very few cartilage. And now let's come to the first is the skull. Okay. So the skeleton is divided into two parts. One is called the axial skeleton. Axial skeleton is your skull and your vertebral column with your ribs. Okay. And after that, there is appendicular skeleton, which is the limbs, your two hands and two legs, which come out from the center axis. So first, let's talk about the axial skeleton. So axial skeleton has 80 bones out of 206. So you can realize our axial skeleton, which is the head and our spinal cord and the ribs, they just contain 80 bones. So majority of bone is present in our limbs, in the hands, shoulder, hands, the thighs, legs. Okay. Is it clear? Great. So axial skeleton has 80 bones and it contains skull, vertebral column, sternum, and ribs. So remember that. Underline skull, vertebral column, sternum, and ribs makes the axial. Remove the limbs and whatever is left is the axial skeleton. It forms the body axis. Write down. Axial skeleton forms the body axis. Okay. Now, first thing is skull. Now, if you look at skull, the skull bones are divided into two parts. One is what forms this cranium. This, can you see my cursor? Hello, both of you? Thumbs up, yes. So this small box, not so small, it's a big box as compared to other organisms. So this box where our brain sits is called cranium. So bones are either called cranial bones or these bones which form our face are called facial bones. Okay, so there are two types of bones in our skull, cranial and facial. Cranial bone means the bone of the head, the cranium, which contains a frontal bone, a parietal bone, temporal bone, occipital bone, and this occipital condyle. Okay, and also sphenoid bone. Sphenoid bone also comes in your cranium. From there onwards, you see that there is a mandible, which is the lower jaw, and this is the only movable bone in the skull. So in our skull, the only bone that we can move is our mandible. Write it down. Mandible is the only movable bone. So our lower jaw is movable. We cannot move our upper jaw. It's fixed. It's fixed with the cranium. Then sphenoid, ethmoid, lacrimal, nasal, all these are like, lacrimal is where your tear glands are. So the bone beneath it at the corner of your eyes. Nasal bone is your nose cavity. Do you see this part? So this part, the outer nose, is all made up of cartilage. So the bone part is just like this. So that's why whenever you see a skull, a bony skull, you will not see a nose on it. Because nose is not made up of bone, it is made up of cartilage, the outer nose. Is that clear, everyone? So far, so good? Okay. So the cranial and facial total makes 22 bones. Cranial bones are 8. And the important thing about this cranial bone is 1, 2, 3, 4, and then 5, 6, and there'll be on the other side 1, 7 and 8. Okay. So this temporal will be on the other side as well, right? So there will be 8 total cranial bones. When a baby is born, the boundaries of these cranial bones are not properly set, and these bones are not very hard. So that's where babies are more prone to fractures in their skull. That is where in their skull are very soft. So you should never hold a baby or make a baby sleep just on a bare, flat, hard, rigid surface with this head bone, because these bones are softer when a baby is born, and these boundaries are not very properly sealed. Okay, we can't see it because there is a skin on the top, there is a scalp, so we can't see it, but that's the thing. These 8 bones gets hardened over time after the birth. Okay, and the face is made up of 14 skeletal bones. Total it becomes 22. Okay. Now coming to some neat questions, what they can ask is, what are the smallest bones in our body? So the smallest bones are the ear bones, inner ear bones. Okay, sorry, middle ear bones. So in the middle ear, right here, you have 3 bones called malleus, incus, and stapes. It's called MIS, MIS. These are the smallest bones of our body. Right down. Malleus, incus, and stapes are the smallest bones in human body. Okay, so head is clear. That's all you needed to know in head. Now let's come to vertebral column. Our vertebral column is total made up of around so it says that the vertebral column is formed by 26 serially arranged units called vertebrae and is dorsally placed. Okay, but if you so 7 are cervical, which are present in our neck, and this is conserved. So note it down that there are 7 cervical vertebra in every mammal, whether it is a human, a chimpanzee, a mouse, a giraffe, any mammal. They have 7 cervical bones. Then there are 12 thoracic bones. This can change because a snake has a very long, some organisms have very long thorax also, but cervical bones are mostly in mammals conserved. Also thoracic to many extent is conserved. So 12 thoracic bones, 7 cervical, and 5 lumbar. Okay, it makes 24. Lumbar is till your end of the waist. After that, there are a sacral and a coccygeal bones. So this coccygeal bone is called coccyx, which is the remnant of a tailbone. Right now, coccyx is the remnant of the tailbone. Okay, is it clear? Give a thumbs up if it's clear. So coccyx is the remnant of the tailbones in humans. Organisms which have tail, in those organisms, this coccyx continues to grow out and forms the tail. And this remnant of a tailbone in humans suggests the evolutionary link of humans to the tailed mammals. Okay, so humans and chimpanzees don't have tails, but they have this final end bone, which is the remnant of a tailbone. Monkeys have a fully functional tail, and there this coccyx grows to form the tail. There can be rare disorders where you will see that some human babies are born with some tail-like appendage coming out from their lower back, but that will not have bones. Okay, because the gene that leads to this tail formation is not active in humans. Okay, it's active in monkeys and other tailed mammals. So what you need to know about vertebral column is the vertebral column has 26 total bones. The thoracic vertebra or the thoracic region where there are 12 bones, there are also bones extended out which are called ribs. So these 12 thoracic bones are connected to the 12 pair of ribs. Write it down. 12 thoracic bones in the vertebra are connected to the 12 pair of ribs. So the vertebral column is at the back side, the dorsal side. In the front side, what protects our chest region is the ribs. And then in the very front, just where we wear a tie on our shirts, is this sternum bone. It is a flat bone in the midline in front, in the ventral thorax. Ventral is front, dorsal is back. Okay? Clear? Devangi, Jani? Very good. So the 12 pair ribs associated with 12 thoracic ribs. So all these 12 ribs, let me go to rib here now. Yes, you see, all these 12 ribs are at the back side connected with the vertebral column. Okay, 12 thoracic vertebral columns. But on the front side, when they come, so this is sternum, which you see in the center, like a tie. This is sternum. At the back, there is a vertebral column. But only 10 out of these 12 are truly connected to sternum. Can you see that? If you count 1, 2, 3, 4, so these 7 are connected completely. So they are called true ribs. 7 out of 12 are called true ribs. The last 3, 8, 9, and 10 are called false ribs, because if you see properly, 8, 9, and 10 are not connected directly to the sternum, but they are connected through the 7th rib here. Look, they are joined here on both sides. Can you see that? Thumbs up if you can understand what I'm saying. So for the first 7 ribs, there is a 1-to-1 attachment. They are attached at the back and also attached at the front, back, front, back, front. The 8th, 9th, and 10th are together attached with the 7th and then attached to the sternum. So they're called false ribs. And the last 2, 11th and 12th, are not at all attached to the sternum. So they are called floating ribs. So our last 2 ribs, which are most prone to fracture because they are floating, and you can feel it. Even if you want to feel your own ribs, just keep going down and you will see that where you're in the center of where your sternum ends, your ribs still go ventrally, like as you go towards dorsal, you will see your ribs, and you will feel that the last 2 ribs are actually floating. They are not attached to sternum in front. Clear? So the axial skeletal is clear. Skeleton is clear. Devangi, Jania. True ribs, false ribs, floating ribs. Why they are called so. Now, the bones of the limbs along with their girdle. So there are 2 girdles in our body. A shoulder girdle from where our hands originate, and a hip girdle from where our legs originate. Okay. So this is the shoulder girdle. So this is the shoulder bone called the clavicle. Okay. Joined to clavicle is the scapula. And these 2 form a joint here, which is called from where the humerus is there. Then there are 2, radius and ulna. Then there are carpals, metacarpals, and phalanges in our palm. So you can see a lot of bones are present in our hand, much more than the total hand. So in your palm, there are much more bones. So you have 8 wrist bones, 5 palm bones, and 14 finger bones, finger bones. So for this together, you will see that there are 27 in number, right? Is it clear? So 27 bones are only present in your palm, where you wear the gloves, that's it. From this point to this point. Then 1, 2, and 3 are present in the whole hand, arm, total making it 30. So there are 30 bones in every limb, 30 in each. So there are 2 hands and 2 legs. So how many total bones are present in hands and legs? 120. Okay. Just in our hands and legs, there are 120 bones. And how many bones were present in our central axis? Just 80. In our head, spine, and the ribs, total 80. And just in our hands and legs, 120. Is it clear? 80 plus 120 is 200. How many bones did we had total? 206. So rest 6 bones form our girdle. So where are these rest 6 bones? 1 clavicle, 1 scapula here. On the other side also 1 clavicle, 1 scapula. 4. And then in this region, you have this big coxal bone on both the sides. 6. Okay. Cool. 206 total bones we counted. So this is all, and then you have to read about joints. So I'm giving you the type of joints and this disorder of muscular and skeletal system as a homework. These are just facts. So just quickly going over it, there are kinds of joints which are fibrous joints that do not allow any movement. So there are some joints where there is no movement. For example, our skull. Remember I showed you that skull bones are joined together, like a stitched together. There is no movement possible between these bones. So they are called fibrous joints. Then there are cartilaginous joints, okay, where cartilage helps, where bones are involved and joined together with the help of cartilage. So between every vertebra here, you see all these 26 vertebral bones are joined together with cartilage in between. Okay. So see here. So this is the bone, the white part is cartilage, bone, cartilage, bone, cartilage. That kind of a joint is called a cartilaginous joint. There is a very limited movement. So we can bend our knee, we can stand straight, we can go a little back towards on the back side also while doing yoga and exercises, but the movement is limited. We cannot make our backbone move a lot. Then the third kind of joints are synovial joints, where a fluid-filled cavity is present between the bones. And this allows a lot of movement. So all our locomotory joints, for example, the joints between our shoulder girdle, pectoral girdle, and the humerus is called a ball and socket joint. So we can use our hand. We can do a much wider, broader movement with our hand. We can throw a ball, we can throw something. It's ball and socket joint. Then knee joint is called hinge joint, and then pivot joint, gliding joints, and also on. Okay, just read about these. And finally, just read about these disorders. Myasthenia gravis and muscular dystrophy are the major ones that are asked a lot. Arthritis, itis means inflammation, arthro means joint. So arthritis is inflammation of joints. Osteoporosis means, osteo means bones, porosis means small holes, pores. So osteoporosis is an age-related disorder where the bone mass decreases and bone becomes light in weight and hollow, and there are more chances of fractures. So you will see that older people can get fracture even if they fall from a chair, whereas young people don't get that fracture very easily, right? Have you experienced that? Have you seen that? Older people are more prone to fracture because of this condition called osteoporosis. So as you grow old, your bones become less and less dense. Okay. Cool. And gout is an inflammation of joints, and that's all. So this chapter is over. And in the next class, we'll be starting with a new chapter. And so I think we have finished the human physiology part, which is part of your syllabus. So now we'll go to the plant physiology.